Today we start the chapter on enzymes, and this is syllabus code 5090, and we are going to be studying O-level biology, for which we are going to be following the 2021 syllabus, and we are going to examine all the details which we need to know about the chapter on enzyme. This is of course chapter 3 of the syllabus, and we will discuss it in details, the first syllabus first, and then we will continue with it. Every student must first go through the syllabus. This is the syllabus, as you can see. This is chapter 3, which is enzymes. Now we have to study enzyme action, we have to study effects of temperature and pH. So the first point is define catalyst as a substance that speeds up chemical reaction. It is not changed by the reaction. So first we define catalyst. Then we define enzymes as proteins that function as biological catalysts. So part A of the syllabus, part B of the syllabus. Then explain enzyme action in terms of the lock and key hypothesis. And the last one is investigate. Now, whenever there is an investigate, please remember, we need to be knowing the practical details of this. So how would we do these in the lab? Because this is what is expected of you, is that you would be doing lab work and studying the effect of enzymes, and you'll be studying the effects of temperature and pH on enzyme activity. So first of all, how do we measure enzyme activity? We've got to know that. Then we have to study the effects of temperature. So if we change the temperature, only then we can study its effects. And then we've got to study the effect of pH. So we're going to change the pH and study the effect on the rate of enzyme activity. So I would always advise you to look at the syllabus before you start the chapter, and then look at the syllabus again after you have revised the whole chapter to see if there's any point which is not clear to you. So this is a study skill. Please remember, there are study skills which you must have. And then there are some which are called exam skills, and we'll be talking about those in a separate video of the exam skills which are needed for you to be doing your biology exam. The first point was, what is a catalyst? You study catalysts in chemistry, like for instance, I don't know much of chemistry, but I think I know is iron is a catalyst. And I think one of the students said vanadium oxide. So I'm sorry, I don't know much of chemistry, but these are probably catalysts. Now these are catalysts, but they're metals. So catalysts, what do they do? You study catalysts in chemistry, what do they do? They speed up chemical reactions, but they're not used up in it. So they remain unchanged. We can say they remain unchanged. We can say they're not used up. So they can be used again and again and again. Now we say enzymes are biological catalysts. Now, for instance, when you think of biology, you think of just two things. I've often seen from our students, they say, Miss, a plant is, we study in biology, we study humans in biology. So we just study plants and animals. Now, I disagree with that. Because you've got to understand is that when we are saying biological catalyst means that they are found in all living organisms. Now, what living organisms come to your mind? Fine, there's a plant. You look out of the window, there's a tree. So fine, plants. Then you look on the wall, there's a lizard. Then you look in a pond and there are frogs. So animals. But then what about the yogurt that you had for lunch? That contains bacteria. So what am I trying to tell you is that enzymes, when you think of enzymes, you've got to think, where are they found? They're found in plants, trees, a rose bush, a mango tree, an apple tree. They're found in animals, a lizard, a monkey, a cockroach, a frog, a gorilla, an elephant. They're found in the yogurt that you had for lunch. So bacteria. Then they're found in the mushroom that you had in your pizza. So fungus. Then what about the pizza bread? How was the dough made? The dough had yeast in it. So the yeast, which is a unicellular fungus, and then mushroom, which is a multicellular fungus. So you can see it in plants, you can see it in animals, you can see it in bacteria, you can see it in fungus, and you can see it in protocysts. Protocysts are unicellular, like protozoa, like amoeba, like plasmodium that causes malaria. So what you have got to understand is that, when you think of enzymes, now in a plant, photosynthesis take place, requires lots and lots of enzymes. In animals that we study, respiration needs enzymes, digestion needs enzymes. In bacteria, enzymes are released and they digest food outside. Fungus does the same thing. Protozoa have enzymes in them, which of course function. So when you think of enzymes, please remember to think of it as in the entire of the biology. And biology is made up of two branches of biology exist. One is called botany, and the other is called zoology. Zoology is about animals, and botany is about plants. Some of you don't like studying a lot of botany, but it's interesting. Don't sort of say you don't like botany, because then when you say you don't like something, you will definitely not like it. But if you be negative about something, then you'll remain negative about it. So I'm sure botany is as interesting as zoology is, but it's the mindset which you make that botany and plants is difficult. So if you have that mindset, well, then you will make it difficult for yourself. Now, the points that come to my mind when I am thinking of enzymes is number one, they're made up of proteins. So if they're made up of proteins, then means they're made up of amino acids. Then we say proteins, enzymes are specific. Now, why are they specific? Because what they have on their surface is this depression. And this depression is called the active site. So the depression on the surface of an enzyme is called the active site. Depression, or we use the word cleft. So the depression on the surface of an enzyme makes it very specific, because only a specific substrate can fit in it, which has a shape complementary to the active site. Then we say all enzymes have their own optimum temperature. So there are certain enzymes, like human enzymes, they would have an optimum temperature of 37 degrees Celsius. But a bacteria which survives in volcanoes would have an optimum temperature, very high optimum temperature. So every enzyme has its own optimum temperature. Every enzyme has its own optimum temperature because every enzyme is from a different organism. We humans, our enzymes work best at 37. Then every enzyme has its own optimum pH, meaning optimum means that it works best at that temperature or pH. Now the word which we use very often is that enzymes are denatured. Now the word that you use is killed. We say enzymes are killed. Enzymes are killed at high temperature. Now that is absolutely wrong. You do not use this word enzymes are killed. Enzymes are denatured. Denatured means that the shape of the active site is lost. Shape of the active site is lost. So if the shape of the active site is lost, then the substrate no longer fits and the reaction is not going to proceed. So we say proteins. Number one, enzymes are proteins. Number two, enzymes are specific. Number three, every enzyme has its own optimum temperature. Number four, every enzyme has its own optimum pH. And then we talk of enzymes being denatured at temperatures above their optimum. So enzymes are denatured at temperatures above their optimum. So if an enzyme has an optimum temperature of 37, it'll probably be denatured at 50. If an enzyme has an optimum temperature of 100, it'll probably be denatured at 130. So please understand that enzymes will be denatured at temperatures above their optimum, and whatever their optimum is. So you can't say all enzymes will be denatured at high temperatures, because if an enzyme's optimum temperature is 100, well, it's going to be denatured above that. So please understand that the word denatured and the word optimum, how has that got to be used? So enzymes are denatured at temperatures above their optimum. Now here you can see there are four different enzymes. Now what is different about them is, number one, they all have different shapes of the active site. So the active site of this one is here. This is the depression or the khadda in it. Here, this is the depression or the cleft in it. Here we have, this is the depression or the cleft in it. And here, this is the depression or the cleft in it. So different enzymes have different active sites. So the thing that I have colored in green is called the active site. Please do not say site. Some of you write site at times, and that would be very wrong. Now, what happens is when a substrate which is complementary to the active site fits into it. So this would be the substrate for this. The substrate. Please get the spelling right. It's substrate. Now this substrate can break into two, or two can be joined into one. So we say this initially, when enzyme plus substrate, we call this enzyme substrate complex. Please just remember, this is not Asan chappal store. It's enzyme substrate complex. Enzyme substrate complex. And then what happens? After this, we have the enzyme is free to be used again. And what do we get? We get product. And why do I write S in a bracket? Because at times it's one and it's time two products. So that's why I'm writing the S in brackets. So enzyme plus substrate is enzyme substrate complex. And the short for that is ESC. And then finally we have enzyme plus products or product. So we have to be clear on these terminologies that we talk about: enzyme plus substrate, enzyme substrate complex, and then we have enzyme, and the final whatever is could be one or could be two, and this product or products. Now how do we study the rate of enzyme action? The rate of enzyme action can be studied. Let's look at a very important and a very interesting example which we talk a lot about. Is the first thing that we talk about is starch. Like you eat bread, so the starch is digested to maltose, and we need an enzyme here which is called amylase. So if we are studying this experiment in the lab, we can study two ways by which we can measure the rate. The first method is, how long does it take for the disappearance of the substrate? So say there were 1,000 starch molecules. How long did it take for them to become zero? Well, maybe it took 3 minutes. In another situation, it took 5 minutes. In another situation, it took 7 minutes. So naturally, this would be the fastest one because it only took 3 minutes and the entire starch was digested to maltose. So how do we study the rate of enzyme action? How long did it take? So we say, how long did it take for the disappearance of the substrate? This is one way that we study the rate of enzyme action. The second way by which we can study the rate of enzyme action is how fast is the formation of the product. So say in the beginning, maltose was 100. We somehow measured that. And then it was 200. And then it was 500. And then it was 1,000. And then it remained 1,000. What does this mean? It means now no more starch is left. The maltose is the final amount of maltose is 1,000. So it's not going to increase after that because all the starch is now zero. There's no starch left. So no starch and this is the total product that was formed, is the total amount of maltose that was formed. So formation of product. So if this whole time took place in 3 minutes, or it took place in 5 minutes, but if it took place in 3 minutes, that means this was a faster rate of reaction. So rate of enzyme is studied on number one, disappearance of substrate, and number two, formation of product. Now let's look at the next syllabus point, which is the lock and key mechanism. In the lock and key mechanism, what you have to understand is that the enzyme is the lock. So in the reaction that we're going to study, I'm going to be discussing two reactions. Starch is converted by amylase into maltose. So amylase is the enzyme. Please remember ASE. This is what we write with every enzyme, this word ASE, amylase, maltase. So the word amylase is the enzyme. Now that is a lock. Similarly here, the maltase is the lock. So the lock is amylase. So this is the lock and this is the lock. Now what is the key? The key is the starch, because that is the substrate which is going to be formed. So the key is starch, and in this situation the key is maltose. So the same maltose which was the product in the first reaction now is the key in the second reaction. This is the key, this is the key. So what we need to understand is that the lock and key hypothesis means that just like, we have a lock and the key fits into it because the shape of the key is complementary to the substrate, to the lock. So in different reactions, you have to remember is that the enzyme would be the lock and the substrate would be the key. And of course, these of course are classified as the product in these reactions. But then the same maltose becomes the key here. So please understand, in different reactions, the same molecule could be labeled something else. So, but your understanding of it must be correct. The lock is the enzyme. So the lock is the enzyme and the substrate is the key. Now let's understand the effect of temperature on the rate of enzyme action. So we have the rate of enzyme action here on the y-axis. We have temperature in degrees Celsius on the x-axis. Please remember this is the independent variable, so we are going to change it and we're going to take different temperatures and study the effect of it on the rate of enzyme action. Now I've asked you to use the terminology of arbitrary units. Arbitrary units means that we have given some random units to it. Like I would say, okay, the best burger is 10 and the worst burger is 1. How many marks do you give to McDonald's or to Hardee's? And somebody might say 7, somebody might say 8, somebody might say 1. So it's arbitrary units. Arbitrary units means I have given it some number 1, 2, 3, 4, 5, 6, 7 or 10, 20, 30, 40. Arbitrary. There's no unit, there's no millimeter, there's no milligram, there's no kilogram. So the temperature is plotted on the x-axis and the rate of enzyme action is on the y-axis. Now you see, as you increase the temperature from A to B, the rate is increasing slowly in the beginning and then it increases. And then we see the rate decreasing. So from A to B, the rate is increasing, B to C, the rate is decreasing. And you can see the optimum temperature of this is 30 degrees Celsius. As we have increased the temperature above 30, the rate has started to decrease. Now at times we ask you a question and I say describe A to B. Please remember, describe is no biology. You do not give me any biological explanations. You will only give me points which are relevant, like you would say, At 5 degrees Celsius, you have to give me the units. The rate is 10 arbitrary units. At 10 degrees, at 15 or 20, better so 5 it is 10. At 5 degrees Celsius it is this. At 10 degrees Celsius it is 20 arbitrary units. So you're just going to read off the graph and give me figures. Now what you have to understand is that describe, you don't give me any biological explanations. And what we find is that you at times write it, and then for the explain you give me what you're supposed to give me in the describe, and I can't give you any marks. So describe is different and explain is different. So what you have to understand is that what you write in describe is very specific. The answer must be specific to the graph. At 5 degrees, this is the rate of enzyme reaction. At 10 degrees, this is the rate of enzyme reaction. At 15 degrees, this is the rate of enzyme reaction. So you will give me figures for that, and that will get you your marks for this part, which is the describe part. Ask you to give me explain A to B. Now in explain A to B, you are going to give me a biological explanation, which is increasing temperature. Increasing temperature increases kinetic energy. So it is going to increase the kinetic energy. Now when the kinetic energy is increased, what happens? More collisions. Some of you say more successful collisions or more fruitful collisions. And this will result in more product formed per unit time. More product formed per unit time. So when you look at the describe was just telling me increasing temperature increases the rate, and you just gave me some figures of the graph. But in explanation now, you have to give me actual biology if you know biology. If you don't know biology, well, you can't do this part. So increasing temperature increases kinetic energy. So more collisions. More collisions means more enzyme substrate complex is formed. More enzyme substrate complex is formed. So more product formed per unit time. So the rate increases. This is what you would write in explain A to B. Now in describe B to C, what happens? Increasing temperature decreases the rate. So you give me some figures from the graph that at maybe you can say 35, this was the rate, and then as it is increased, then what was the rate? So you give me figures from the graph. And this would be the describe B to C. Explain B to C. If I say explain B to C, you've got to give me, now you're increasing the temperature. Increasing temperature above the optimum. Now this is important. Increasing temperature above the optimum is also increasing the kinetic energy, but that is not good for us. It starts partial denaturation. Partial denaturation means not complete denaturation, but it's partial denaturation. Slightly the active site is starting to change its shape. So say this was the enzyme and this was the active site. Now there's a slight change in the active site, but still product is being formed. So still product is formed. That is why we call this partial denaturation. Rate is decreasing. So product formed per unit time is less. product formed per unit time is decreasing, and at C is complete denaturation. Complete denaturation, all the active sites are now have lost their shape and the substrate no longer fits. So you understand what is happening between B and C. As you see, between B and C, the rate is decreasing. So at between B and C, the rate is decreasing. But what is happening is that the shape of the active site is slowly. Now a time will come, it will be totally lost. So the substrate will no longer fit. This is another diagram to show you the effect of temperature on the rate of reaction. And as you see, temperature increases, the activity increases, and then of course a point comes. This is the point of the graph. And then the denaturation decreases the activity, and you can see the percentage active of the enzyme. So this is a graph which I just wanted to pause the video and have a look at it and see how at different temperatures how the rate is increasing and how the graph after the optimum. So this would be the optimum. This would be the optimum, and after the optimum then there's a very short, this is the only range at which it works. And at this point the enzyme is totally denatured. Now this is a very nice graph which is depicting an enzyme is less active at very low temperatures. Now this is the important thing is that for every 10-degree rise in temperature before the optimum, the rate doubles. So as the temperature rises, enzyme activity increases as indicated by the increase in the rate of reaction it catalyzes. Usually the enzyme is twice as active for every 10-degree rise in temperature until the optimum temperature is reached. So before the optimum, so say if this was 10 degrees and this was 20 degrees, so the rate here would be 50, the rate here would be 100. So for every 10-degree rise in temperature, the rate doubles. So this is what is considered in most of the enzymes. This is the situation. The optimum temperature reached, enzyme is most active. Beyond the optimum, enzyme activity decreases. At point D, the enzyme has lost its ability. So what we need to understand is that the effect of temperature on the rate of reaction. Now this is a graph showing the different pH. So you can see pepsin has a pH which is around 2, salivary amylase has a pH around 6, and alkaline phosphatase, another enzyme, has a pH around 9. So different enzymes have different optimum pH, and you can see it's a very narrow range. And enzymes would be denatured at temperatures above their optimum pH. So before that, they would of course remain inactive, but as the pH is nearing its optimum, they work their best. And if we increase the pH, please remember pH is what? Power of hydrogen. So this side is acidic and this side is alkaline. So what we need to understand is how every enzyme is different. So pepsin, the one which is present in our stomach, has got the lowest pH. And why? Why? Because we have HCl in the stomach. And the HCl is important in the stomach because when we eat food, it contains a lot of bacteria. And those bacteria will be destroyed by the HCl and also by the pepsin, because if you remember, bacteria have cell wall of peptidoglycan. And peptide is a protein. So you have to remember is that the pepsin and the HCl is actually protecting you. We will continue with the remaining part of this chapter in the second video on this chapter. And thank you for now.